Okay, so let's talk a little bit about muscle physiology and muscle tissue. A lot of information in this chapter. It's going to take me several uh, days to get through this, so I want to start it now. I'll break these up into some short, manageable videos, but this is an awful lot of information. So, overview of muscle tissue. So, there's three types of muscle tissue. We talked about these earlier in the semester. There's skeletal muscle, there's cardiac muscle, and there's smooth muscle. Um, the generating force or muscle tissue is the basic function of all muscle tissue. Muscle tension is the basic function of all muscle tissue. As I said earlier when we were talking about tissues, the main role of muscle tissue is movement. I'm going to cross in front of you right now to see if I can get a little better view from the other side. Yeah, this is a little better. I'm in a new room. I haven't used this room before for making any videos, so this is all trial and error. So hopefully I can get back in our regular room at a later date. Other functions of the muscle tissue, again, create movement, maintain posture, stabilize joints, generate heat, and to regulate the flow of materials through hollow organs, such as your esophagus and your, I'll think of the word I'm looking for here in a moment, your esophagus and your intestines. Skeletal muscle tissue is made up of long, multinucleated cells. They are arranged parallel to one another. They extend the entire length of the muscle. They're known as fibers due to their length and appearance. So each muscle fiber is actually an individual cell. Contractions of skeletal muscle are voluntary. We control those. We can control whether we pick something up or set something down. Most are attached by connective tissue to the skeleton, ergo the name skeletal muscle. Contraction can produce movement of a body part. You contract your biceps brachii muscle and your arm moves like that. There is the figure from your textbook showing the skeletal muscle. Here's the cell. There's the multiple nuclei, the long cylinder striations of the muscle fibers, mostly attached to the skeleton again. They are voluntary and they produce movement. Cardiac muscles are found only in the heart. They are short and highly branched with one and at most two nuclei. They have a structure called an intercalated disc that join adjacent cells to each other. They contain gap junctions and desmosomes, which are modified tight junctions. They unite cells and permit coordination of contraction. When the heart muscle contracts, it contracts completely and then relaxes completely. Contraction of the heart muscle is involuntary, not controlled by conscious thought. We do not control how often our heart beats with our conscious thought. And then there's a figure from the textbook. See one nuclei, one nuclei, one nuclei. This is one cell. This is a cell. This is a cell. You see the branching of the cells. Everywhere you see an intercalated disc, that's where the cells are connecting. Found only in the heart for cardiac muscle. It is involuntary and it produces the beating of the heart. Smooth muscle has no striations. You go back a couple of views, see the striations in the skeletal muscle. You see the striation in the cardiac muscle. In the smooth muscle, we will not see striations. The cells are going to be long and flat. They all have pointed ends. That type of shape is referred to as a spindle. When something is sort of pointed at the end and then bulges in the middle, that is referred to as a spindle. Single centrally located nuclei. They line most of the hollow organs. They're also found in the eye, the skin, and some of the ducts of certain glands. They are also under involuntary control. Many are linked to one another by gap junctions. These gap junctions allow for a synchronized contraction. Again, that is where you can't see me very well. Let me step into the light. A synchronized contraction will be one area contracts. This area doesn't contract yet, but this area contracts. And as this area starts to contract, this area will start to relax. So what you have is a motion that looks like this. That's the way food moves through our digestive system, through a synchronous motion. Again, there are figures from the book. You see the cells pointed at the ends, bulging in the middle, centrally located nuclei, found in the walls of the hollow organs, and again, under involuntary control. Changes the diameter of the hollow organs, causes the hairs to stand erect, adjusts the shape of the lens and the size of the pupil of the eye. So there's an overview of all three types of muscle tissue: skeletal, cardiac, and smooth. See all their associated differences. These two have striations, this one does not. These have multiple nuclei. These all have central nuclei and one. These may have up to two. Most of them are only going to have one. The big thing about these is this branching pattern that all of these cells show. Properties of muscle tissues or muscle cells. All muscle cells have contractility, which is the ability to contract. The proteins in the cell draw closer together. Does not necessarily involve shortening the cell. Excitability is the ability to respond to some stimulus. The stimulus is some signal that comes from outside, such as a chemical, a mechanical stretch, or a local electronic signal. And they all have conductivity, which is the ability to conduct those electrical charges across the entire plasma membrane. Later on in this chapter, when we start talking about membrane potential, the conductivity is what leads to membrane potential. Extensibility is the ability to be stretched up to 3 times their resting length without rupturing or being damaged. And elasticity is the ability to return to the original length after stretching. When we think about the elastic in your sweatpants, we know that you can stretch that, but the role of the elastic is, yes, to stretch, but to bring it back to the original size once it has been stretched. Structure of an individual muscle cell. This is our generalized cell from chapter 2 or 3. We have the plasma membrane, the cytoplasm, the smooth endoplasmic reticulum. There's the mitochondria and the nucleus. In a muscle cell, we have many of the same components, but we name them a little bit differently. Again, here's the nucleus, here's mitochondria. We have, instead of all these other components, we have a muscle fiber, a myofibril right here. The sarcoplasmic reticulum is surrounding the myofibril, takes the place of the smooth endoplasmic reticulum. The cytoplasm has now been replaced by what is called the sarcoplasm, and the plasma membrane has been replaced by a very similar structure, but a structure known as the sarcolemma. So we have, and an individual muscle cell is called a myocyte. So as I move on through this lecture and I start using the term myocyte, I am referring to a muscle cell. The myocyte has, again, the sarcoplasm, which is the myocyte's cytoplasm. The sarcolemma is the myocyte's plasma membrane, and the sarcoplasmic reticulum is a modified endoplasmic reticulum. It forms a web-like network surrounding the myofibrils, and it varies in structure in the 3 different types of muscle tissue, and we will talk about that later on as we talk about those different types of tissues in more detail. Myofibrils are unique structures found in all muscle cells. They are cylindrical organelles, approximately hundreds to thousands in each myocyte. About 50 to 80% of the volume of the cell is going to be myofibrils. The diameter is about 1 micrometer, or about 1 1/100th the thickness of a human hair. If you want to know what a micrometer actually is, I think we've all seen a meter stick. I don't see one in this room, or I would show you a meter stick. Yep, okay, there's one walking in front of you again. All right, so here's my meter stick. Meter stick, 1,000 millimeters long. So if you look closely, you cannot see this, so don't try. But from my finger to the end of that stick is 10 millimeters. If you take a single one of those millimeters, so if you look across the top of a meter stick, every little mark is a millimeter. If you took the time to count them from one end to the other, there'd be 1,000 of them. If you take that millimeter and then divide it lengthwise into 1,000 pieces, each one of those 1,000 pieces is a micrometer. A micrometer is 1/1,000,000th of a meter. So if we took this meter stick and lengthwise sliced it into 1,000,000 pieces, each piece would be a micrometer. The diameter, they're made up in bundles of specialized proteins which allow for contraction. Again, if we go back to our picture, there is the modified sarcoplasmic reticulum, and each one of the little dots in there represents a myofibril. Again, each myofibril contains thousands of myocytes. Other organelles, such as the mitochondria, are packed between the myofibrils. So let's go back to our picture. See, there's the mitochondria being packed in. There's the nucleus sort of shoved out to the outside. Smooth muscle cells myofibril arrangement is different in cardiac and skeletal muscles, and we will talk about those, I'm sure, when we get a little further along in this chapter. Again, structure of the muscle cell, again, this is just a review. If I'd have known it was here, I would have simply gone forward 1 instead of backward 4, but there is the basic structure of all muscle tissue. All muscle cells look like this. Again, the smooth ones have a little bit different arrangement of these structures, but the basic items are all the same. Skeletal muscle fibers. Let's talk about those. So here's my skeletal muscle fiber, long tapering fiber, multiple nuclei. We see the nuclei, we see all the striations. So skeletal muscle fibers consists of skeletal muscle tissue, consists of many fibers and surrounding endomysium, which is the extracellular matrix. The fibers are thin cylinders. They're approximately, or can be up to 30 centimeters long. Red blood cell below is only 7.5 micrometers in diameter. So there's your red blood cell, so 7.5 micrometers, and there's a typical muscle cell. So again, back to our idea of the cell from early in the semester, we remember that cells are individual units, but they can vary greatly in size. The thickness of these can be up to 100 micrometers. Again, here is a muscle such as the biceps brachii muscle, where it's attached to bone by a tendon. Here is the entire muscle. That is the blow up of the muscle fiber right there. So there's a whole bunch of fascicles. There's an individual fascicle. There is a fiber. We'll talk about fascicles again in a moment. But here again is the blow up of the individual fiber, showing the sarcoplasmic reticulum, the individual myofibrils, structures called T-tubules or transverse tubules. That's where materials come through and out. There's the openings into the T-tubules. The fibers are formed by fusion of many embryonic myoblasts. Remember, blast refers to an immature cell, giving fibers multiple nuclei. So you have many of these will come together to form a single a single fiber. The myofibril is the most abundant organelle. It's made up mostly of contractile proteins. The sarcoplasmic reticulum surrounds the myofibrils, stores and releases calcium ions. Later on, we will talk about calcium. The transverse tubules are found deep inward extensions of the sarcolemma. They surround each myofibril. They form a tunnel-like network within the muscle fibers. They are continuous with the exterior of the cell. They are filled with extracellular fluid. Again, there are those T-tubules right there. They contain a terminal cisternae, which is an enlarged section of the sarcoplasmic reticulum. These structures flank each T-tubule. Two terminal cisternae plus the corresponding T-terminal form a triad. There's the triad right there. There's the cisternae on one side. There's the cisternae on the other side. There's the opening of the tubule forming the triad. There's the tubule, and there's one cisternae, and there's the other. Structure of an individual myofibril. Each myofibril is made of hundreds to thousands of myofilaments, consisting of one or more types of proteins. Contractile proteins generate tension. Regulatory proteins dictate when a fiber may contract. And structural proteins maintain proper myofilament alignment and fiber stability. So think about, these are the ones that cause the movement. These are the ones that control whether it's moving or not. And these are the ones that form the framework. There are three types of myofilaments. There's thick filaments and thin filaments and elastic filaments. The thick ones are bundles of a contractile protein called myosin. Thin filaments contain the protein actin, along with tropomyosin and troponin. The elastic filaments have single massive spring-like structural protein called titin, which stabilizes the structure and resists excessive stretching. And again, here are those items. There's the myofibril, there's the structure, there's the thin filament, there's the myosin tails, here's the thick filament, or excuse me, this is the thick one, this is the thin one. I should actually read what I'm saying. And it's just the way these things will blow up. This is a thin filament, but you see it's bundled into these myosin tails, which is here. And then we have the thin filaments here. Thick filaments contain, again, the protein myosin. They have globular heads on this end. At each end, it's linked by intertwining with the tails of the thin filament. The heads are connected to the tails by a hinge-like neck. That's this structure here. And each head has an active site that binds with actin. Here is the actin. That's where the heads of the thick filaments are going to bind. Multiple actin subunits string together to form two intertwining strands in a functional thin filament. Each bead-shaped actin has an active site, binds with the myosin heads. The tropomyosin is the long, rope-like regulatory protein. That's the sort of light blue one that's holding everything together. And then the troponin is the small globular regulatory protein, excuse me, which holds tropomyosin in place and assists with turning contractions on and off. Ultimately, what happens is the way a muscle contracts is these heads will attach to the active sites. The hinge will hinge up, pulling this forward, and that will contract the muscle or shorten the muscle, and then they will release, which then allows those structures to go back. This is what I refer to as a Z-disc. It's where everything is coming together. There is the overlap zone here. There's the M line. which is the myosin structure, and then there's another Z disc over here. Ultimately, it's this part of the muscle right here that is going to stretch and get bigger and then return. Myofilament arrangement, microscopically, striations are alternating light and dark bands with specific regions. The I band is light. It's mnemonic, only found in thin filaments, I in light. Z discs are in the middle of an I band. They are composed of structural proteins that anchor the thin filaments in place and to one another. The Z disc also serves as attachment points for elastic filaments and attach the myofibrils to one another across an entire diameter of the muscle fiber. The A band, A dark, that's another mnemonic, contains the zone of overlap with both thick and thin filaments, generates tension during contraction. AJ, again, mnemonic. H is in the A band, the middle of the A band where only thick filaments exist. The M is in the middle, that's the M line. It's a dark line in the middle of the A band. Structural proteins hold the thick filaments in place, serve as anchoring points of the elastic filaments. And there we go, the I band here, in the H band, the H zone. There's the zones of overlap, and then the outer edge of the A band. Big picture, multiple muscle fibers, each surrounded by an endomysium, form a fascicle. Each fascicle is surrounded by a perimysium. Bundles of fascicles make up a skeletal muscle surrounded by an epimysium. So we have an endomysium, forms fascicles, fascicles bundled together by the perimysium. Bundles are the skeletal muscle surrounded by an epimysium. Perimysium and epimysium come together at the end of the muscle to form a tendon that binds the muscle to its attaching structure, which is most often bone. Skeletal muscles are enclosed by a thick connective tissue layer called a fascia. The fascia anchors them to the surrounding tissue and holds groups of muscles together. If you've ever deboned a chicken, for instance, and you started trying to pull the chicken muscle away from the bone, that sort of white layer of tissue that's holding the meat very tightly to the bone, that is the fascia that we are referring to. And again, there we start down here with the individual myofilaments, thick and thin, put a bunch of them together to form a myofibril, put a bunch of myofibrils together to form a muscle fiber. Each individual one of these is called a fascicle. Put the fascicle into, the fibril into the fascicle into the muscle surrounded by the epimysium. And that is the big picture for muscles. Now, let's see, okay. There is a type, we're hearing about this in the news, it's been in the news recently. It's a particular type of muscular dystrophy. For those of you who are old enough to remember, I'm trying to visualize the class that I have currently. I think there's probably a couple of you old enough to remember when Jerry Lewis, a comedian, had a telethon every Labor Day for muscular dystrophy. Muscular dystrophy is any type of disease where muscles are wasting or not functioning properly. They're not forming, they're formed, but they don't work. All of those things are muscular dystrophy. Duchenne muscular dystrophy is a very particular type of muscular dystrophy. It is a degenerative muscle disease found almost exclusively in boys. It is caused by a defective gene for the protein dystrophin, which is coded on the X chromosome. So Duchenne muscular dystrophy is in fact an X or a sex-linked disease. Dystrophin is the structural protein in striated muscle. It anchors the sarcolemma to the surrounding connective tissues and to the myofibrils. So basically, in this type of muscular dystrophy, since this dystrophin is being disrupted, the muscles don't hold together, and they come apart, and they won't function that way. There was a young man that had this, was a big fan of the Harry Potter books. He was on Oprah Winfrey a couple of times. He has since passed away from his muscular dystrophy. I think he also had a brother that had it. Again, he was on Oprah Winfrey. I think his name was Maddie, I believe. In absence of normal dystrophin, the sarcolemma breaks down, muscle fiber is destroyed, replaced with fatty and fibrous connective tissues. The symptoms usually appear between the ages of 2 to 12. They include weakness of the proximal limb muscles and the waddling gait. Generally, these people are wheelchair bound by age of 12, and they pass from cardiac or respiratory or cardiac failure by age 20. The young man I was referring to that was on the Oprah Winfrey Show a couple of times, again, he was a big fan of the Harry Potter books. He was wheelchair bound much sooner than 12, and I don't think he lived to be 12 or 13 years old. Excuse me. These are showing normal muscle fibers. This is what a normal muscle fiber works. This is what the muscle fibers of someone with Duchenne muscular dystrophy would have, both magnified 260 times. And you see the fat deposits being replaced all through here that are replacing the muscle tissue. All right, we're going to stop for right there for the few minutes, let my throat rest, and then we will pick up with the sliding filament mechanism of contraction.